Hi everyone, my name is Augustin Hennings and I am a fourth-year graduate student working in the Dunsmore and Lewis-Peacock laboratories at the University of Texas at Austin. So this project begins with the idea of encoding specificity, which was first introduced by Thompson and Tolving. This is the process in which neural circuits which encode a memory reactivate during retrieval to guide behavior. So the research of learned fear and subsequent extinction has been able to demonstrate this principle to great success in rodents. And we now know that fear and extinction memories exist as stable and separable engrams in rodents. But how can we study these processes in humans? We don't have access to the level of neural circuits that engrams have been observed at in rodents, but we can use an analogous approach called encoding retrieval similarity. Instead of individual neurons, we can correlate spatial patterns of activity as measured by fMRI from encoding to retrieval. This correlation is the degree to which these two states overlap. We have a very specific hypothesis of where we expect neural reinstatement of fear and extinction memories in the human brain. Previous research in animal models suggests that different areas of the prefrontal cortex code for memories of opposite valence, and there is converging evidence from human research that suggests the same is true in humans. Specifically, the dorsal anterior cingulate cortex, or DACC, codes for fear memories, and the ventromedial prefrontal cortex codes for extinction memories. However, most of the work in humans has only used univariate measures and has not looked for neural reinstatement. This brings us to our specific research question. Are associative emotional memories organized the same way in humans as they are in rodents? Our specific hypotheses are that there will be a double dissociation of emotional memory reinstatement in the PFC. And secondly, individuals with PTSD will display dysregulated reinstatement of emotional memories, consistent with PTSD phenotypes of being unable to recall extinction memories. So in order to test our hypothesis, we employed an episodic associative hybrid task. In this task, participants formed an emotional association to a semantic category, either animals or tools. However, each individual stimulus is a unique category exemplar. For example, there is only one type of dog shown throughout the entire experiment, only once. This means that each stimulus is encoded in a distinct emotional context based on the type of associative learning taking place. The task starts with a baseline phase in which participants are passively viewing the stimuli. During fear acquisition, half of the images from one category are paired with a mild electric shock to the wrist. Extinction immediately follows fear acquisition, during which no shocks are delivered. The following day, participants are placed back in the scanner and complete a surprise recognition memory test for the items they saw the previous day. The end result of this task is that each episodic memory has one implicit emotional association, none, fear, or safety. So in order to measure the amount of neural reinstatement, for each item we correlate the patterns of activity from encoding to retrieval, as shown on the right. So we are interested in the amount of emotional reinstatement. We take the difference of the CS plus and CS minus items to control for the amount of overall reinstatement elicited by seeing the same stimulus twice. So this brings us to the main results. First, looking at healthy adults. On the y-axis is the CS plus minus CS minus neural reinstatement, and on the x-axis is the encoding phase. As a sanity check, we see that in both the DACC and VMPFC, there is no significant selective emotional neural reinstatement for baseline items. For fear acquisition, there is significant emotional reinstatement in the DACC, but not in the VMPFC, these purple bars in the middle. The opposite is true for items encoded during extinction, the green bars. We see significant emotional reinstatement in the VMPFC, but not in the DACC. So the story is not the same for individuals with PTSD. There is significant emotional reinstatement for items encoded during fear acquisition in the DACC. However, there is no significant reinstatement for any encoding phase in the VMPFC. Jumping to group comparisons, we see that healthy adults are exhibiting significantly more selective emotional reinstatement in the VMPFC for items encoded during extinction. So, in conclusion, we observe that healthy adults exhibit a double dissociation of associative emotional memories in the PFC, and individuals with PTSD display dysregulated selective emotional reinstatement. We hypothesize that this distinct organization of emotional memories in the PFC may be adaptive in healthy adults, such that it may prevent memories from being overwritten. If you have any questions or would like more information, please feel free to send me an email or to use the live chat during the poster session. Thank you so much for watching.